Thank you for the invitation from ASM and also opportunity for me to talk about my research in this FAScinate talk series. My name is Lim Po Teen, an associate professor from Institute of Ocean and Earth Sciences, Universiti Malaya. I'm specializing in harmful algae. CSI was one of my favorite TV drama series. For CSI's fan, you probably could recall the interesting twist and turn in the drama series before the complete were reviewed from the crime scene with close examinations of the physical evidence. Back then, I was always amazed with the advance of instruments used in every episode. In today's talk, I would like to share my research in harmful algae, of how we have unveiled or discovered killer algae in our waters, and explain what harmful algal bloom or HAB is all about. And we will dive into different type of HAB event that affect us in different way, including how it make people sick by producing various potent neurotoxins, and what we have learned throughout the years. And I will emphasize why precision biodiversity in HAB is crucial. When we say about algae, many of us will think about seaweed, profila, or in Japanese, the nori, that we use in making Japanese sushi roll, or the health supplements such as spirulina that many are using. These are examples of useful or good algae that widely used as food or health product for public. If we collect a drop of seawater, we will have the chance to see the amazing microscopic world with living creatures ranging from few micron to few hundred micron in size that we can see the shape or form under high magnifications of microscope. These microscopic algae are the base of trophic level in aquatic ecosystem. They are just like terrestrial or aquatic plant with photosynthetic pigment that can utilize sunlight, carbon dioxide, and produce organic material and become the food for zooplankton, fish larvae, and form the core or basal of the marine ecosystem. With the increasing nutrient loading from the applications of chemical fertilizers, organic waste from households or agricultural industry, our coastal become very much enriched in nutrient. This will in turn promote the growth of selected algae species, for example, dinoflagellate or diatom species, eventually forming algal bloom. When these algal species are harmful, it will cause negative effect to our coastal ecosystem. Harmful algal bloom are very diverse in nature. Different harmful microalgae species responsible for different type of shellfish poisoning, as you can see in these particular photos. The most common type of shellfish poisoning are listed here. And the most common one in our region is what we call the paralytic shellfish poisoning. Shellfish become the vector of human poisoning when the shellfish are contaminated and become toxic. Some species are known to be responsible for massive mortality of fishes or shellfish, both from the wild or in the culture species, and responsible for huge economic losses. The impact of HAB also vary from years to years, region to region, and with different in natures, mainly cause problem in public health or economic losses. The impact of HAB to tourism or other sectors was not clearly accounted for. In the state of Sabah, at least 200 cases of shellfish poisoning, mainly paralytic shellfish poisoning, have been recorded and with 20 casualties. The emerging HAB event in Johor in 2014 alone responsible for at least 600 tons of fish mortality in a single HAB event. So if you are interested to look at the HAB event and the trend of HARP in Malaysia. So we also have prepared a report under the Global HARP Status Report that was published for the couple of years. Over the 30 years of HARP in Philippines and Malaysia, what we have learned. In the IPCC Special Report on Ocean and Cryosphere in a Changing Climate, several statements were given to highlight harmful algal bloom and the consequences due to this phenomena. Harmful algal bloom display range expansions and increased frequency in coastal waters since the 1980s in response to both climatic and non-climatic drivers. The observed trend in HARP are attributed to partly to effect of ocean warming, marine heat wave, oxygen loss, eutrophication, and pollutions. HARP or harmful algal bloom have negative impact on food security, tourism, local economy, and public health. The first type of shellfish poisoning I would like to share here is what we call the paralytic shellfish poisoning, or PSP. This is a type of shellfish poisoning due to contaminations of group of neurotoxins called saxitoxins. First PSP event in Malaysia can be dated back in 1970s in Sabah, and it remained the seafood public safety and public health issues due to frequent occurrence of algal bloom species in the state. With a systematic monitoring program in Sabah that put in place, it helps to prevent human poisoning due to algal bloom. In Peninsular Malaysia, Alexandrium minutum was first identified after a poisoning event reported in year 2001 when 6 school children were hospitalized, including 1 casualty. This was the first record of the species and was part of the study that I involved directly when I was a graduate student in UKM. Recurrence of bloom of this particular species continues since 2001 with different levels of impact. This is a photo taken during the bloom in 2015 that cover almost the whole lagoon of Sungai Geting, Tumpat. The toxin level in clam exceeded 40 times the safety levels. With the early detections and confirmations, a shellfish ban was issued and no human poisoning was reported in the area. This is an example of result from chemical analysis of toxins by using HPLC. Because all species produce a different suite or combinations of toxin composition was used to confirm the source of the toxins and with the toxins found in the toxic vectors. The toxin profile can be used as a fingerprint to confirm who the toxin producers. In the event of harmful algal bloom or shellfish poisoning, shellfish collections and ban will prevent the contaminated clam from getting into the market. Of course, the business of the local trader, fishermen, shellfish collectors, or some restaurant operators in the area will be affected with the ban. In the management of HARP, efforts should be placed to ensure public health and at the same time should be balanced to minimize the economic impact. This is crucial to help the livelihood of the local traders. This is the data we obtained from a 4-month study of bloom of Alexandrium minutums in 2015. The upper graph is the cell density of the toxic species, while the lower graph is the toxicity in the clam. Our result clearly show the relationship between the toxic bloom and the toxicity in the clam in the area. We also clarify the underlying mechanism and the relevant environmental factors that induce the occurrence and formations of the bloom of this particular species. Our finding help the relevant monitoring agency in the management of algal bloom event in the affected area. The second type of algal bloom event related human poisoning that I would like to share here is what we call the ciguatera fish poisoning. Many species of reef fishes, for example, barracuda, snapper, and grouper, or eel, become not safe for consumption or vector of poisoning. This is a type poisoning occur when the toxic algae from the genus Gambidiscus become high in abundance in the reef ecosystem and attach to seaweed and other form of substrate in the coral reef ecosystem. When they are grazed by small herbivorous fishes, then get into the carnivorous fishes, the toxins bioaccumulate and biomagnify into the trophic levels and higher food chain. Some marine invertebrate, including giant clam, sea urchin, were also reported previously as the vector of ciguatera fish poisoning. The compilations of newspapers cutting here clearly highlight the severeness of fish poisoning or ciguatera fish poisoning. But this type of poisoning was not given enough attention in Southeast Asia regions until very recent years. You can clearly see the number of cases reported worldwide, with the number of poisoning estimated between 50 to 500,000 cases annually. One of the unique symptoms of ciguatera fish poisoning is what we call the dry eye sensations or the burning sensations when the victim tries to hold a glass of water or drinking the glass of water. Unfortunately, the hotspot of ciguatera and the mechanism how to monitor this type of poisoning continue to be a subject of research, with many technical challenges remain. Our most recent work related to ciguatera fish poisoning case was in Sabah in 2017. You can see the chronology of the event here. This is the first chemical confirmations of ciguatera fish poisoning case in the country. The fish flesh collected from the victim's house were confirmed by genetic methods, and the toxin in the flesh was confirmed by LC-MS. This case has become a very useful references among the medical practitioners with the report and confirmation of case in Sabah. Please refer to the articles by Lee et al. In Malaysian medical journals for more details of the case. Thanks to the funding support and from True Science Fund and International Collaborative Fund from the Ministry of Science, Technology and Innovations for this work, we have the funding and opportunity to conduct benthic hub research in coral reef ecosystem in Perhentian Island. In this study, the higher toxin producers, which is the Gambidiscus, and the cell abundance were detected in microhabitat dominated by tough algae associated with coral rubber fragment, 86% of total samples in zone 2, the one highlighted in red box. This is in agreement with many studies which reported benthic substrate with tough algae hosts the highest density of the toxin producer. To keep the story short, what we learned from this study is degradation of coral reef will certainly alter the benthic hub ecosystem and promote the growth of toxin producers Gambidiscus species. So we can protect our coral reef and prevent or minimize the risk of ciguatera fish poisoning. Anaphylactic shellfish poisoning is another type of shellfish poisoning I would like to share here due to contaminations of vector shellfish with a neurotoxin called domoic acid. ASP has neurological and gastrointestinal symptoms. ASP was also known to cause permanent loss of short-term memory in the victims. ASP is not only a public health concern, but also was reported to cause mass mortality of seabirds and marine mammals in other regions. So precise identification of these diatom species require the use of transmission electron microscope up to 100,000 times of magnifications to confirm some of these ultrastructural features at submicron levels. This is crucial to differentiate the species that are known to be toxic from the non-toxic species. We can only manage what we know, and precise and correct identification of toxic species is critical. Over the last 10 years, we have confirmed at least 30 species of pseudonyxials with descriptions of 9 novel species and new species from different parts of the country. And we can clearly see here, this is not a localized problem. and toxic diatom are widely distributed in our waters. In this graph, you can see the red dot, that the locations that we have samples to confirm the presence of toxic diatom, and precise identification of these species, especially the toxin producers and their distributions, is very, very important with the rapid growing of aquaculture area. This type of data will certainly help us to better prepare in dealing with this type of harm event and potential public health issues. We also discover and confirm another toxin producer from our mangrove and coastal ecosystem. This is how they look like under light and transmission electron microscope. Here are the common edible bivalve in Malaysia. Most of these clam and snail were collected from coastal, tidal flat, and mangrove forests. Based on our studies, some of these samples that we collected locally were found to be contaminated with dormant acid at trace levels. And the DA level will be within the regulatory limit of 20 milligram per kilogram. In this study, the toxicity of benthic clam, benthic diatom, nixia were confirmed by both HPLC and LCMS analysis. So we also know the toxicity vary among the strain analyzed. The DA was detected as the major congenant in most of the strain, and the blue dot line clearly indicate the toxicity of each cell actually vary from one strain to another. Each number represents one of the cultures in the lab that we have tested for dormant acid that we have established from different parts of the country. All our data provide strong indications that ASP and dormant acid should be included as part of the national HAB and shellfish monitoring program. In most of the emerging harm incidents, the impact was most severe due to species that without prior record, or in area that without harm monitoring program. For example, the massive fish mortality in Johor in 2014 and 15. Our aquacultures, as I mentioned earlier, our aquaculture suffer severe damage with 600 tons of fish killed in a single event. Is there anything we can do to detect the presence of this harmful algae species in our coastal water? The answer is yes. With the rapid advancement in molecular techniques, including DNA, environmental DNA, and next-generation sequencing metabarcoding, we can assess the diversity of phytoplankton, including harm species. Hundreds of samples were collected from research crews with UMT discovery since 2015, and these samples were analyzed. This allows us to confidently confirm the occurrence and distribution of harm species in our waters, as highlighted in the graph. And a total of 28 harm species was confirmed and annotated from the sequence reading, with 7 new records in Malaysian waters. We also observed higher numbers of OTU, the Operational Taxonomy Unit, of harm species, mainly along the Malacca Strait. This data will be useful for mariculture industry to assess the potential risk to shellfish poisoning and also the threat of fish killing algae to our mariculture industry. We also developed species-specific quantitative PCR method that allow us to detect and confirm the selected species in these samples. In this case, the toxic species known to responsible for paralytic shellfish poisoning were detected along the South China Sea and Strait of Malacca. We also assist using the method that we have developed to assist researchers from Mexico to confirm the presence of these species along the Mexican coastal waters in a collaborative study. Our marine ecosystem is changing in response to both climatic and non-climatic drivers. The synergistic impact will have severe consequences on our human and ecosystem health, food safety and security, I hope by now you are convinced that we can no longer ignore the socio-economic impact of harmful algal bloom. Of course, it's never too late to do our part to reduce the anthropogenic pressures, to conserve our ecosystem, the coastal ecosystem, and put in effort to rehabilitate the degraded marine ecosystem. Improvise hub monitoring program but ensure public health, food safety, and food security for our future generations. I also like to take the opportunity to thank all my graduate students that are currently at different universities, and also my international collaborators for their direct and indirect contributions to this work. Thank you very much.